And next, it is my pleasure to introduce Dr. Sujatha Kannan. She's the Vice Chair of Research for our own Department of Anesthesiology and Critical Care Medicine. So thank you so much.
Thank you. I'm very excited to talk to you about the future of translational research in neurocritical care. So I'm also honored to, I have the great honor to hold the Traceman Endowed Chair. And as many of you might know, Dick Traceman was the founder or the father of translational neurocritical care research. Essentially, he was one who, whatever he saw at the bedside is what he tried to replicate in the lab, and that's how this rich tradition has started. And I think anyone who's been working on neurocritical care research has indirectly or directly been linked to his work. What I would like to say is to start is this, where I think the future of translational neurocritical care research is in cellular targeting. So how can we target specific cells in the brain? And I'm going to be talking about some technologies related to that. Now, I'm a pediatric intensivist, and I do pediatric neurocritical care, which is, that's my clinical practice. That's been my practice for the last 10 years or so. Though I trained as a pediatric intensivist. And this is a relatively new field. It's only about 10 to 15 years, and we've been learning from the rich tradition of the NCC program here. So some of my research is going to have a little bit of a pediatric tilt to it. However, as we believe, adults are nothing but spoiled, overgrown little kids, so we can extrapolate from that. The data that I'll present, I'll be talking about some non-FDA approved therapeutic agents, and the patents are all, we have more than about 100 patents related to this technology that are, many of which are licensed to our first startup, which is a Hopkins startup, Ashwatha Therapeutics. This is a clinical stage company now, and we're also co-founders of this therapeutics. Others are licensed to newer startups that are coming up now. And so the conflict of interest is managed by Hopkins. We obviously are entitled to royalty distribution and equity, both myself and my husband, who I collaborate with, and Hopkins. So I'm just going to give an example of a patient that I saw many years ago, and this is kind of what started me in this area. And I think you'll find links to this in what your practice is in the NCCU now. So this is obviously a child, is a 5-year-old who came in with, was completely normal, came in with fever and seizures that was uncontrolled. So was actually in the ICU for about 2 months and coming on and off pentobarb coma. We threw everything at him, the kitchen sink pretty much, steroids, IVIG, whatever you could think of, and nothing worked. The seizures didn't stop. So he was in status every time he would come out of pentobarb. At that time, there was a tracer that was, this was at Children's Hospital of Michigan, where I was before coming here. And at that time, there was a tracer to identify activated microglial cells, which is PK11195. And we used that to identify, and you could not see that area on FDG PET. And we found an area where there were activated microglial cells, which was causing the seizures. Once that was surgically removed, the seizures stopped, and at least he was able to become somewhat functional. So the area that was resected was positive for microglial cells. So this is the immune response was what was triggering these continuous seizures. So this is what I look at in the lab, and we look at inflammatory cytokines that are produced by the microglia. We look at surface markers, but what do these glial cells do? So when you image this, when you look at a live imaging of a brain slice, in a normal brain, this is how the microglia are. They're kind of zipping around, clearing up debris. This is their normal function, so they're very active. And this is a slice from an animal with cerebral palsy, and you can see that these microglial cells are kind of just sitting around, and they're not able to do the function that the regular microglial cells are doing. And this has been linked to many disorders in pediatric, many neurodevelopmental disorders, and also in a lot of conditions that we see in neurocritical care and in neurodegenerative diseases. So can modulating these immune cells lead to recovery and repair? And how can we get to these cells specifically? Now, the question of targeting, what we try to look at is one of the things that has a lot of potential is using nanoparticles, and a lot of people have been looking at that over the last 2 or 3 decades now. But the challenge of targeting is the same if you think about the scale of size. It's the same as throwing a soccer ball in the Arctic Ocean and expecting it to land here. And if you inject a nanoparticle IV peripherally and expecting that to get to the brain, it's a similar scale. So it's very difficult if you think about it that way. What about if you put a ligand on it? And that's something that people have looked at too. So getting through the blood-brain barrier, it's a very crowded space. And this is how, I don't know if anyone's been to Mumbai or you've been to the train station there, that's how the train station looks. So that's packed with people. And if you think about the size scale of it, putting a ligand, which is like a mosquito, on an elephant, and then you make them, you're expecting the mosquito to lead the elephant through this crowded station across. So that's again how, when you think about if you expect a ligand to get to a receptor on a cell that is deep inside the brain to deliver the drug, that's essentially the way the scale works too. So this makes it extremely challenging, and which is why getting things specifically to certain cells in the brain has always been a big challenge. So what are the issues here? We have the blood-brain barrier, which is very difficult to get across. Inflammation is often diffuse. It's not just in one area, it's throughout the brain or in multiple areas in the brain. And even if we can get the drug through, will it accumulate in enough concentration in the cell to have an effect? And the biggest issue is many times the injuries already happen. By the time we see the patient, the injuries happen, there's quite a bit of inflammation that's going on, and can they recover from whatever insult that's occurred to them? So I collaborate with my husband, who's a chemical engineer. I hope there are no chemical engineers here because one of the things that he always says is that chemical engineers can solve any problems, and they are problem solvers. Whatever the problem it is, they can solve it. So this is the problem I took to him, and we've been working on it for 20 years now. And he says, well, chemical engineers are like a blank slate. They always think out of the box, and we can come up with unusual solutions. So one of the things that we started looking at were these dendrimers. So these are 4 nanometer particles, which have many surface end groups, and you can attach drugs, ligands, imaging agents to it. And the way nanoscale particles function in the presence of disease was something that no one had looked at. So that is something that we started looking at to see how it functions when there is an existing neurologic condition or existing disease. So what we try to do is leverage the intrinsic properties of what these nanoscale particles do, and then build on that to make it a delivery device. So what we found over the last 20 years or so that we've been working together is that these hydroxyl dendrimers intrinsically target inflammatory cells. So they are picked up by macrophages and activated or inflammatory microglial cells. They are like water. They're very, very soluble, very hydrophilic, and they function and diffuse through material like water. It does not need any ligand to get to these cells. And we've seen this in multiple models. The only time it's not taken up is you don't have inflammation is not the primary pathology over there. It can be provided either oral, systemic, which is IV, subcutaneous, any intranasal, any of them. As long as it can get into the bloodstream, it reaches the glial cells. It crosses the blood-brain barrier easily, especially if there's some small amount. All you need is a little bit of perturbation. It does not cross a normal blood-brain barrier. So if you're healthy, if it's intact, it will not get through. And the other exciting thing is it's easy to produce. It's very predictable. You know exactly what it does. You can predict the pharmacokinetics. It's always the same. And we've been working on this, and we've had multiple NIH grants for it. Of course, we've collaborated, have had wonderful collaborators throughout Hopkins that we work with. So what are the barriers? One is the blood-brain barrier. Even in the presence of injury, linear nanoparticles like dextran or hard nanoparticles do not get through, while the dendrimer diffuses through and gets into the brain parenchyma. But once it gets into the brain parenchyma, it needs to get taken up by these cells. And that happens very rapidly. Within about 4 hours, the cells which are these activated glial cells pick it up. And we can see this. This is the model that I work a lot with, which is a rabbit cerebral palsy model. And there is a lot of neuroinflammation that happens in the periventricular region. So there's a big ribbon of activated microglial cells in the periventricular area, and that's what picks up these dendrimers. The other exciting thing about it is the greater the inflammation, the more gets into it. So if you have the more severe the injury, the more microglial cells, activated microglial cells you have, the more the uptake. So the disease regulates how much gets in, which is another unique thing because the amount of drug that we would deliver would also get regulated based on how severe the injury is. And we've seen this across multiple models. So in traumatic brain injury, cardiac arrest, optic nerve injury, even neurodevelopmental disorders like Rett syndrome, autism. So we've tested this in multiple species, multiple kinds of disorders, and it's pretty much the same that we see. So the first drug that we tried to look at for efficacy was N-acetylcysteine, and you might wonder why N-acetylcysteine. It's just kind of available over the counter and it's never worked. There have been a lot of clinical trials especially for neurologic disorders with N-acetylcysteine. So if you go on clinicaltrials.gov, you'll get 10 pages of trials that are listed over there and barely any papers or anything that came out of it. And the reason we think, one of the reasons is that the way it gets transported into the cell is through a glutamate antiporter. So for cysteine to get into the cell, glutamate has to get kicked out. And we all know that many neurological injuries or neurological disorders have a lot of excess glutamate. So the extracellular glutamate goes up, and which may be a reason why it never worked. Now, when we put it on the dendrimer, it bypasses all of these mechanisms. It gets phagocytosed into the cell, and then it releases only inside the cell. So the drug can be conjugated to the dendrimer in such a way that it can be released the way we want it to. So in this case, we wanted it to be intracellular release, so it's intact in the plasma, it gets excreted intact through the kidney in the urine if it's not picked up. Whatever is picked up releases over time inside the cell. So we looked at it in my rabbit model of cerebral palsy. We saw that there is improvement in motor function, there's decrease in oxidative injury and inflammatory cytokines. And I'll just show you a short video of how these rabbits look. So these are newborn rabbits that were exposed to inflammation in utero, so they have a phenotype of cerebral palsy. They drag their limbs, they are not able to walk. This is abnormal movement that you're seeing here. And as they get older, this one received a single dose of saline. This one received a single dose of 10 milligrams per kilo of dendrimer N-acetylcysteine. And most of this is a pretty severe model. Most of them die by about day 10. And after a single intravenous dose, you'll see that this one is still not back to normal by about 10 days, but as they get older, they have catch-up growth and start looking more like normal rabbits. So there you can see that this one is getting better. So we've seen this in other models of hypoxic ischemic encephalopathy, other types of brain injury, and we also see that it helps even with stem cell therapies because it decreases the inflammation, and then the stem cells are able to take better. So we've tested this again in multiple models, but when we started thinking about translating it to patients, we were initially looking at childhood cerebral adrenoleukodystrophy, which is a fatal disorder This is seen in boys and by usually about 4 to 6 years of age. And most of the time, by the time they are diagnosed, most of them do not survive beyond 2 years after they are diagnosed. So this is because very long-chain fatty acids are not cleared out. And we see what we did is we took the monocytes from these patients, and we found that they are pro-inflammatory when they're exposed to very long-chain fatty acids, and treatment with dendrimer and acetylcysteine suppressed that inflammation. So this is a trial that we are looking at for, this is a patient population that we're looking at testing it in. Now, our first clinical trial, first in human trial, was in patients with severe COVID. And this was around 2020 when we were getting ready for getting this platform ready for getting this therapy ready for clinical translation. And then when COVID happened and patients had, we had patients with severe COVID who were also having neurologic injury afterwards, this was a patient population that we wanted to look at. And this was a multi-center trial, a phase 2A trial. A phase 1 was previously done in healthy volunteers and had no safety issues, because if it's not picked up, it pretty much gets excreted in the kidneys. The way this was designed is that we patients were stratified into getting three doses. The first is a pretty low dose. If you think about it, it's 2 milligrams per kilo of dendrimer N-acetylcysteine, which actually has less than 0.5 milligrams per kilo of the N-acetylcysteine itself. And so that's like water. We use N-acetylcysteine typically in gram quantities. So it's very little that was given, and we escalated to 8 milligrams per kilo. And each cohort had placebo patients too. Of course, all of this was blinded, and 24 patients were enrolled in the study. And if this is the first bar is the placebo group. Among 7 patients who received the placebo through this 8-month period, this is a single dose. So patients were given a single dose at the time that they were diagnosed. These were patients who were in the ICU, were intubated, and were classified as severe COVID. So they would get 1 IV dose of the dendrimer and acetylcysteine at whatever dose they got stratified to get. And then they were followed for up to 60 days. So in the placebo group, we only had 2 patients who were alive and ventilator-free at 60 days. While you can see that with each of the treatments at 2 milligrams per kilo, we had 2 patients who died, nobody was on a ventilator. 4 milligrams, everyone did great, and at 8 milligrams, we had 1 patient who died, but that was because they withdrew support. This was an elderly patient where support was withdrawn. So we can see that there is we felt this was a pretty impressive improvement clinically. So this correlated with a really nice decrease in CRP. This was a time when patients were getting steroids and remdesivir. Tocilizumab was not in, and baricitinib, all of those were not part of practice at that time. So there's a very nice increase in CRP that you can see in these patients too. So the CRP comes down faster and much more rapidly. The really exciting thing that I felt that came out of this was that the brain injury biomarkers came down. So we had a nice decrease in neurofilament light, GFAP, and tau that was seen in these patients, in the serum of these patients. This has not been described with any of the other therapies that has been tried for COVID. So this indicates that the therapy gets into the brain and gets to the activated glial cells and is able to suppress the brain injury biomarkers. So this was very exciting. And Aaron Gustin, was actually the first author on this paper. So this is very nice work that was done. We've tried other therapies, of course. We have about 80 different small molecules that have been conjugated to the dendrimer and have been tried in different kinds of disorders. The other drugs that we work very closely with the Slusher Lab, that is the Translational Drug Discovery Group at Hopkins, And they have many interesting compounds that are extremely potent and toxic, so these are perfect for attaching to the dendrimer because we can prevent the side effect profile, so the side effects profile is completely taken out. So we've shown that targeting glutamate carboxypeptidase is effective in an EAE mouse model. Glutaminase is another inhibitors are another very potent compound which cannot be translated clinically because it causes sloughing of the gut when you take it, just because glutaminase is present everywhere. And again, this is something that can be attached to the dendrimer and delivered, and we do not see the side effect profile. What we have also shown is that these specifically inhibit the pathways only in the cells that they're delivered to. So when we have taken out glia and non-glial cells, we see that it inhibits it only in the inflammatory glia and not in the other cells. So which again narrows down the off-target effects a lot. So what is the next? So we have clinical trials that are ongoing with these, with this platform. So what is the next thing that we are looking at? Now we have a different kind of dendrimer that has been created, which is kind of the next gen dendrimer is what we are calling it. These specifically target hypermetabolic neurons. So these are made of glucose. So neurons that are hyperactive or hypermetabolic pick it up because they are faked into thinking that it's glucose. It does not degrade or get metabolized, but it can deliver drugs into these cells. And typically, seizing neurons are ones that would be hypermetabolic too. Again, injured neurons at different stages of injuries would be hypermetabolic. So we showed that these are taken up by neurons, but the hydroxyl dendrimer, the other platform, does not get into these neurons. So we've shown that both in vitro and in vivo, and it can be delivered. We've looked at it intracranially and intranasally, and in both cases, it goes into these specific neurons that are, and this is in a seizure model, so these were in neurons that were seizing. We attached it to a simple drug, valproic acid, and showed that we were able to increase the latency to seizures in a pilocarpine model. It also decreases high-grade seizures, stop status epilepticus. So this is just very early data. So this is a very new platform that we've just started working on. So what are the other things that we are also working on? One of the other things that we are doing is delivery of siRNA. So gene editing and microRNA, siRNA, different gene editing tools is what we are now delivering. And again, this is a great platform to specifically manipulate certain cells in the brain, not everything, which makes it very attractive. And this again, is a proof of concept study where we showed that Cy-GFP can be delivered to tumor-associated macrophages in a glioma model. So what are the current applications that are being explored? We are looking at clinical trials in adrenoleukodystrophy, in cerebral palsy, and cerebral palsy from various causes, including hypoxic ischemic encephalopathy. There is an imaging. This can be used to image the neuroinflammation in the brain, and this is being developed as a diagnostic along with a therapy, and that's currently in phase 1 trials. There's a phase 2 trial for a systemic therapy for age-related macular degeneration. Current therapies for age-related macular degeneration is pretty much an injection into your eyeball to get it into the vitreous. To target the retina. But this is systemic subcutaneous therapy, and this is a trial that's ongoing. It's an open label trial, and it looks very promising. These are all the other indications that we are looking at, and indications such as subarachnoid hemorrhage, stroke, ICH, these would be good targets to go after for the neurocritical care population. So to conclude, what I think the future is precision nanomedicine, and cellular targeting is probably where the next range of therapies should be. And these platforms can be used to modulate the CNS immune response It is scalable, it is very predictable, and it can be personalized based on what needs to be treated or what pathway you want to go after. And it's because it is very predictable, it's easy to switch different drugs on it or change it to what you exactly need for the kind of therapy that you would need it for. The other thing also is gene therapy. Right now, there are a lot of therapies, RNA and gene therapies that are coming up, but the issue is delivering it to the right cells. And this platform provides that, that possibility. And that is where, again, I think the future of treating many neurologic and neurodegenerative disorders would be based on this. But again, the precise targeting is what would help for it. So as I mentioned, the ongoing clinical trials now are the phase 2 for AMD and phase 1 for ALS. And this is something that I've learned, that it is critical for those who are working at the bench and in the bedside to be involved to make sure that the translation happens the right way. Otherwise, it takes on a life of its own and doesn't get to the patient population that we really want it to get to. And of course, this is a huge collaboration throughout Hopkins. We work with the Drug Discovery Center for Nanomedicine, Ophthalmology, Kennedy Krieger. We work with them a lot. And of course, pediatrics, neurology, all of these different groups. So these are the different groups that I work with. Of course, a lot of folks in ACCM, the Center for Nanomedicine, Translational Drug Discovery Group. I work with surgery, Kennedy Krieger. All of this work till now has been funded through NIH. Thank you.